Hi, I'm Chance.
And I'm Sarah Catherine.
And this is Conservation Connection,
presented by Last Chance Endeavors.
We run a wildlife education nonprofit focused on connecting students to their environment.
Each week here on Conservation Connection, we do just that by introducing you to the groundbreaking science and conservation work that's happening every day around the country.
We talk to professionals in the world of conservation science and wildlife management and ask them about their career,
their current projects,
their wild and crazy stories from the field,
and everything in between.
Join us each week to discover just how these dedicated people are working to protect our planet. Today, we're talking with Dr. Erik Sotka. He's a professor of biology at College of Charleston, and he does lots of amazing work here at the Grice Marine Lab.
Welcome to the show.
Thank you. I'm really happy to be here.
We're happy to have you. Could you give us a little bit more on your background of what work do you do here in Charleston?
So I'm an evolutionary ecologist. I think about how marine organisms interact with their environment, and that means not only what they eat and what eats them, but also how they deal with the seawater salinity and the temperature and everything else that they have to deal with. But I also think about how they rapidly evolve in some cases to be able to deal with changes in their environment. And what's great about living and working right on the water is I can go out in between classes and pick up whatever organism I'm working on and be able to play with them in the field or bring them back to the lab.
That's really awesome.
Pretty nice to be able to just step outside and be like, here's my laboratory. This works really well.
It is. I fell in love with marine biology at a marine lab, and so I've always tried to be at marine labs whenever I possibly could. And I was lucky enough to live and work near one.
Makes sense. Yeah. So what's that story? Fall in love at a marine lab?
Yeah, so I'm actually a transplant from Oregon.
Okay.
I grew up in the Pacific Northwest, went to school at University of Washington, and the University of Washington has a marine lab in the San Juan Archipelago, which is in the northwest of the contiguous 48. It's about as far northwest as you can get before you reach Canada. And they have a marine lab there called the Friday Harbor Labs. And I was taking a class on marine zoology, which is all the animals, and a class on phycology, which is all the seaweeds. And at some point during that 10-week semester, I looked up and I saw the snow-capped Olympic Mountains in one direction. I saw the snow-capped Cascades in the other direction. And I'm staring at the water where I see these comb jellies, this group of very odd jellyfish that I was really fascinated by. And I just thought, this is it. I'm getting paid eventually to be in a place like this, hopefully. Studying these beautiful organisms and being in such a fantastic setting. So that's where I said I wanted to be a marine biologist. And I feel lucky because that moment came when I was 20 years old, and I've been doing it ever since. So, yeah.
When it's right, it's right.
Yeah. I think for me, what I do feel lucky because I know that some people have a more tortured path and for reasons that make sense. And sometimes you just luck into what you want to do when you're young. And that's what happened to me.
That's awesome.
Yeah. It's really nice to be in a place where right at the beginning of your career, you look up and you go, yep, This is where I want to be. So that's where you started. Let's talk about where you are now. So what's some of the current research that you're doing in your lab?
So we have been thinking about introduced species for a while now. And what's fascinating to me about introduced species is it's right at the nexus of ecology and evolution.
Absolutely.
So you have species that are here because we artificially brought them here. Sometimes we brought them here intentionally. Sometimes they come because they like our boats, or they come because they hitchhike with things that we bring intentionally. But, for every 10 species that comes, most of them are going to die. And that's a fascinating question. We actually don't know why it is that 9 out of 10 species that arrive just don't make it. But one does. And if it does, it sometimes has a big enough impact to change the local ecology, maybe it makes a new prey base for the rest of the food web. Maybe it makes its impact through being a predator. Maybe it makes its impact by having new mutualisms with local species. It integrates itself in the local food webs in ways that I find really interesting, because they all happened in the last century. They might have even happened in the last 10 years. And everyone's adapting to each other in a way that I find really fascinating. So ultimately, that's what I'm interested in is why does this work for some species? Why didn't it work for other species? And what are the ecological and evolutionary consequences of that?
So something that I found really interesting that you just said is oftentimes when we hear about invasive species, we're thinking like, oh, wow, there's this lionfish and it's this invasive species and it took over. And every kind of species that comes in that's invasive, they just take over and cause all these problems. But what you said was that there's so many more invasive species that are introduced, and some of them, lots of them, just don't make it. So
Yes,
that's
right.
really fascinating. And that's kind of what you're studying, right?
That's part of what I'm studying. It's hard to study what's not here.
Yeah.
Right.
Right. And so I think that's why that's been such a vexing question for invasion biology. How is it that 9 out of 10 species don't make it? To get data on that, you need to do experiments on things that aren't here.
Yeah.
And ethically, that's not probably a good thing to do, right?
Right.
We don't want to start dragging species to a place. What we can do, though, is rely on historical records of things that we know were attempted to come
We're
here.
here at some point.
Yeah, so the one that we've been focusing on recently is an oyster called Cresostria gigas. It's from Japan. We know that it's from Japan because we took the DNA out of this oyster and we analyzed its sequence. And we compared it to oysters that occur in China and Korea and Japan. And we know that it came from northern Japan. And it expanded from northern Japan to every continent on the earth except Antarctica. It's
wow.
everywhere. We brought it for aquaculture.
Okay.
So we brought it because we wanted to raise oysters in Spain and France and the west coast of the U.S. and Chile. And it's still being exported for those same reasons.
Wow.
We also tried to bring it to the east coast of the U.S. There was a huge effort in the 40s, 50s, 60s, through the 80s of bringing this species from Japan that had been so successfully aquacultured everywhere else in the world. But it was dying every time we threw it into the east coast of the U.S.
Huh.
Wow.
So that's a case that's interesting because it's one of the few failures that we can really document. And so we can learn a lot from doing that.
So have you solved that one yet? Do we know the answer? Because I'm really curious
Yeah.
to know, like, why didn't it work?
So no.
Good answer. Well, we'll have to follow up
We'll have
for
to
when
follow
you do
up.
know.
That's right. I mean, there are some places where it was also introduced where it failed because it was too warm. So they tried to
Okay.
bring it to Costa Rica. And northern Japan has the same climate as Massachusetts.
Okay.
So a little different
Right?
from
So
Costa
pretty
Rica,
cold.
maybe.
That's right. So yeah, so that one made some sense, but it was really baffling to the biologists in the 70s about why it wasn't making it. It could either be some local predators that attacked it, local parasites that attacked it. The climate seems to be about the same, so it's really a mystery.
So when you're studying something that's not here,
Yeah.
looking for evidence of an organism that was here for a short period of time or was only introduced and then died very shortly thereafter,
Yeah.
what are some of the other ways that you can read history's logbook of what has been here? What
Right.
other data do you look
I like
at?
that. I like history's logbook. So in the case of the oyster, what we can do is look at all of the other species that came with the oyster. So everyone who's picked up an oyster knows that it's very rough, and you know that the surface has all kinds of nooks and crannies that things can live in.
Right.
And when you pick up a cluster of oysters, there's even more places where things can live and hide in. And we weren't very careful when we first started bringing these oysters about cleaning out those cavities.
Right.
So, not surprisingly, little snails, little pieces of seaweed, little sessile invertebrates that aren't very well known to most people, things we call bryozoans and tunicates and all these other fantastic animals that I love to study. They also were in those little pockets. And it turned out when you brought them here, while the oyster died, everything else survived, or not everything else, but a bunch of those things survived. So they hitchhiked a ride to the east coast of the U.S. So even though the actual vector, the oyster, isn't here, all of those hitchhikers are, or many of those hitchhikers are. And so we can study those hitchhikers to understand that original movement. The two we've been focusing on are a seaweed, which I'll call Gracilaria, and that is one that is now currently from Georgia all the way up into New Hampshire. It's probably expanded into Maine at this point. And that originally came from populations in Virginia, which is where the oysters were first brought. Introduced, that's right.
So that really complicates, not only are you looking at the effect that the original introduced organism has on the ecology of wherever you drop it,
yes,
but you're also looking at the effects that the parasites that were on it or the algae has on that same environment.
That's
so it's really confounding, in science
Very
you want to figure out what single factor caused this.
That's right.
And when you've got 6 things that happen all at the same time, it's really hard to lay out.
Yes.
It's this, not that.
That's right. Yeah, and with introduced species, the big question about how they got here is something like aquaculture versus other vectors that we know are also important. So it turns out that animals and plants can hitch a ride on ships, or they hitch a ride on the inside of big ships. There's a big issue with what we call ballast water. Ballast water is the water that a major ship, and we're talking about the ones that are 2-3 football lengths in size.
Huge.
They take up water to create stability in the ship. So if they're in Japan, they take up water in Japan, and then they drag all that water from Japan, they drag that water to Charleston. And just before they get into Charleston Harbor, they dump the water. But what they've done is transplanted everything that's inside that Japanese harbor, or just outside the Japanese harbor, as in more recent years they've been doing. They dragged all of that water thousands of miles.
And all the microorganisms that are swimming around in it.
That's right. And so that's the other major vector that we see in marine invasive species.
And do they do that basically as a counterweight in the boat so that it's more stable as they travel?
That's right.
That if
that's exactly
one time they're carrying a heavier load or the other time they're carrying a lighter load, the boat moves the same way, right?
That's exactly right.
Okay. Yeah, that can cause some big issues for sure.
And it absolutely has. We know that shipping has accelerated dramatically in the last 20 to 30 years, and we're seeing accelerating numbers of invasive species being spread worldwide because of it. So the ones that I'm studying don't seem to have been brought here through that vector. They've really been brought here through this historical aquaculture
intentional
effort.
means.
And it was brought intentionally. That's right. Although these hitchhikers weren't.
Right. So as you've been looking at this, and as people and scientists in general have been learning more about invasive species and how they get here, how much of an issue it is, have you noticed any change in kind of the regulation of those things? Is there a way that they're trying to stop this from happening?
Yeah. So I would say that we have made some progress on several fronts. So on the aquaculture front, there were some rules put in place in the 70s to make sure that the oysters that were transplanted from one place to the other did not transplant these hitchhikers. So they're improving on that front. And that includes, like, don't move adults, move only the small spat, for instance.
That makes sense.
Or if you are going to move adults, make sure it's not clusters, it's single.
Okay.
And then you clean those off really well.
Very clean, yes.
Very clean, yeah. With ballast water, there are rules now about where you can dump that water and where you can pick up that water. With other forms of vectors, we don't have as effective tools. So a colleague of mine, Amy Fowler, studies bait boxes. So people are moving worms and snails from one place to another. And it turns out that they're still packing these bait boxes with seaweed. And those seaweeds not only themselves can be invasive, but there's all kinds of animals that live in those seaweeds.
Right.
And you know, the seaweed is a great packing material. And I think that's originally how this Gracilaria that I studied came here, was that the oysters were packed in seaweed. And then when you receive the oysters, you know, on the East Coast, you should throw that seaweed up onto the garbage, or you throw that seaweed to dry and on land, but you may just be lazy.
Toss it all
just
in.
toss it next to the oysters, or
Do what's easy.
yeah. So yeah, there have been some common sense practices that have been put in place to deal with invasive species. The problem is that many of them are already here, and once they've established themselves, they're very difficult to get rid of.
What kind of effects are we seeing on, the southeast coastal waterways? What effects are these invasive species having?
Yeah, great question. So I think the poster child for negative effects of an invasive species in the southeast is the lionfish.
Absolutely.
And you all have heard about the lionfish. So it's the lionfish lives offshore on reefs that range anywhere from 30 feet to 200 feet. They are beautiful fish that have these gorgeous spines on the back that are poisonous. They are sit and wait predators. So they'll literally sit in the part of the reef where other fish will be, and the fish will actually be close enough that the lionfish will attack it within 6 inches. But in some cases, all the lionfish has to do is open up its mouth, and these fish are so unaware that this is a predator that they will go into the lionfish's mouth.
Wow.
And they're unaware because it's not something that they normally would have to look out for, because
That's right.
it comes from the Indo-Pacific
Exactly.
and was never in the Caribbean. So they don't have that in their instincts to be like, oh, look, giant spiky fish, stay away from that.
And this is where I find invasive species to be fascinating, even though the impacts are so negative. That fish, all of these fish here in the southeast, did not evolve a behavior to avoid that lionfish. So again, it's this nexus of evolution and ecology, which I find interesting. The fish are doing so well here that they're becoming obese. It's yes, it's the first time that, and I think this is the case, the first time in science that it's been recorded that fish have fat pockets.
Wow.
And so, maybe the hope is that they'll all get some heart disease at some point.
That's incredible.
Yeah.
I had not heard that before. That's amazing.
So that's one highly negative. There's nothing good about the lionfish invader.
Well, they're tasty. They're very tasty, but it's hard to catch them.
But they can be tasty where they're from, not here.
I guess the second one I'd talk about is this gracilaria that I work on, because it is in every high salinity estuary that we find along the southeast coastline, at least from Georgia through North Carolina. It's in every high salinity estuary we've gone into. Now, its effects are more mixed. So seaweeds are at the bottom of the food chain. So there is some benefit to herbivores to having them here. It's a food source.
More food, right?
It also is a place where small invertebrates can colonize. So it's a place where small crustaceans called amphipods and isopods, and then small snails and other critters could get protection from their own predators. And we've actually worked with a decorator worm. So this is a worm that lives in the mudflat, but it makes a tube that extends out of the mudflat about 3 centimeters. And then that tube extends down into the mudflat another foot.
Oh, wow.
And what it's doing is it's constantly gardening its tube. So the little tube that's outside the mudflat, it's cleaning. It's kind of like a stay-at-home dad. It cleans it, basically covering it with whatever's around. And right now what it's doing is it's gluing seaweed to it. So you have this native worm, decorator worm, gluing an invasive species onto it.
Right.
And that invasive species is actually a benefit to the worm. The worm gets some direct food. It actually eats the seaweed. But it also, more probably beneficially, eats all those little critters that are attracted to the seaweed. So it's basically gardening for amphipods or gardening for snails. Now, the seaweed is benefiting as well, because the seaweed is staying at a very quality habitat. It doesn't get washed up onto the marsh, and it doesn't fall off into the depths of our estuary where there's no light. So there's this mutualism going on between this native species and this introduced species. And so that's not all that negative, right? I mean, nothing that I've talked about with this seaweed is negative. The one bit that really worries us with this seaweed, though, is it's changing the nutrient cycle, and it's also changing bacterial loads in the water column. So one of the reasons we don't eat oysters, back to oysters for a second, one of the reasons we don't eat oysters in the summer is because they get loaded up with a bacteria called Vibrio.
Okay.
Right? There's this rule that you have in the Southeast where you don't eat any oysters outside of months with an R in them. So if it's a month with an R, then you can eat the oyster. So you think about that. That's basically the months you can't do it are then May, June, July, August. But by September, there's an R, so you can eat oysters again. Wow. Well, the reasons we don't eat oysters May through August is because of this bacterial load, that there's this huge bloom of this Vibrio bacteria that causes some real negative health effects.
So basically the oysters, which are filter feeders, are filtering the water to get their food.
Yes.
And in doing that filtration, they're pulling bacteria out, and it stays within the tissues of the organism?
It not only stays on the surface of their gills, but they will have the Vibrio living within them, even with the colonization of a very little amount. So It's not just that it's in the water column, although that is an important source. It's also that they have they're host to a whole suite of Vibrios inside their tissues.
And what would eating an oyster that has a high bacteria load, what would that do to people?
So the effects of Vibrio poisoning have to do with attacking our nervous system. People get violently ill, and several people are so sensitive to it that they die to it. So it can be very harmful. Those Vibrio bacteria, and this is the worry we have with the Gracilaria, those Vibrio bacteria are actually thriving in areas where the Gracilaria occurs, where this introduced species occurs. And the reason is actually pretty simple. The bacteria, why do you go to the dentist?
Get your teeth clean.
Yes. Every time you brush your teeth or you go to the dentist, what are you cleaning off of your teeth?
A layer of bacteria that's grown on there?
And why are there bacteria on your teeth? Like, what are you supposed to avoid?
There's lots of food and sugars.
Sugars.
That make the bacteria's life great.
Heterotrophic bacteria love sugars. That's why you get cavities, because those bacteria thrive in your sugar-rich mouth, right? So seaweeds photosynthesize. What that means is they're converting light energy to chemical energy, and they're making sugars as a byproduct. They don't use all that sugar. Seaweeds ooze about half of their photosynthate out into the water column.
Oh, really?
They slough it off. They release it. They don't use half the photosynthate they make, but the Vibrios sure will. So what we've seen with some experiments is that at small scales, when we add Gracilaria to the mudflat, the sediment underneath that mudflat is covered in Vibrio, just huge amounts of it. And we've also seen through a companion study that our colleagues in Virginia have shown is that mudflats with Gracilaria have more of these Vibrio bacteria in the oysters than mudflats without the Gracilaria. So yes, the Gracilaria is having a positive effect for some members of our ecosystem here, but there's this whole microbial ecosystem, this whole community of microbes that are probably being facilitated by the presence of the Gracilaria. And that has some real worrying consequences for human health.
That's really, really fascinating. And I love that, you can tell that you're clearly so passionate about what you do and what you study. In this field, from your past experiences, going back however far you want to go back, has there been just one experience that you're like, man, this is really my favorite experience? This is the coolest thing I've been able to do in this job?
Oh, great question. Well, since that experience when I was an undergraduate, there have been many times where I've wanted to pinch myself. There's no doubt. They all have something in common, which is I am working in a beautiful place in the field. I am in the field. Sometimes it's beautiful, to be honest. Sometimes it's not. But I'm in the field with the organisms and in the ecosystem. And I'm surrounded by invertebrates. And to me, it just makes me whole. It makes me feel like I'm doing something useful that is soul affirming. Whether the two that come to mind are when I'm scuba diving off of South Carolina coast, which I did a lot as a graduate student, 60 feet down where we have nothing but Pleistocene hard bottom reefs, but I'm surrounded by all these fantastic sponges and seaweeds and trying to learn about how those systems work by doing experiments. Or I'm in the Bahamas with a group of undergraduates teaching about marine invertebrates to a group of students that know nothing about marine invertebrates. Whether I'm doing research or I'm introducing naive undergrads to this fantastic diversity, I don't know. It's why I got into it 30 years ago. So
that's awesome.
That's awesome.
It just feels right.
Yeah, it feels right. And like I say, I feel really lucky that I was able to find it.
Cool. Well, speaking of kind of naive undergrads or people who are looking to get into this field, if you had one piece of advice to give to maybe a high schooler or an undergrad who's maybe interested in looking at kind of what you do, what kind of advice would you give them?
That's a great question. So I would say two things. One, stay curious, because you don't know where life is going to take you when you are an undergraduate or a high school student, but you know that it's going to be something that's soul affirming if you're doing something that you're curious about. So that's something that I've always told undergrads and high school students: stay curious. And the other thing I would say on a more practical basis is if you do want to think about doing marine biology or doing science as a career, take as much science as you can. Yeah. Because you don't know whether, let me say it this way, marine biology, you can get a degree in marine biology, and we offer one at the College of Charleston, for instance. But you don't have to be a marine biology major to do marine biology. You need to know a bit about chemistry and physics, as well as physiology and behavior. I mean, you need to know a lot of different things to be able to do it. So try them all out and see what hits you, what strikes your fancy. I guess that's what I would say.
I love it. I definitely would agree. When I was in my undergrad, when I was working primarily with herpetology, reptiles and amphibians. Yeah. But I cannot tell you how many times classes that I took on organic chemistry or other disciplines were really helpful in the research and in the work that we were looking at, or even just the classwork that I was taking. Yeah. And even outside of my undergrad, as we've been figuring out how to start a nonprofit and get other students out into the environment, having a really broad basis is really, really helpful, because just like nature, in science, everything's connected. And
it creates some depth to what you're looking at, too.
Absolutely. And it gives you a basis to understand the broader implications. So not just looking at how this algae has become established, but how that established algae has major effects on fisheries management with the oysters. Looking broader is really where the hard and most valuable work in science is.
I guess the other practical thing I'd say about that is that it may be that you find a career in marine biology, but it may also be that you'll find a career as a geneticist, or you'd find a career as a chemical engineer. It almost doesn't matter what organism or system you study. If you're studying marine biology, but you're learning those other disciplines, then you have the flexibility to do any and all of them.
And that makes you adaptable.
Yeah.
That's a good thing.
It's a good thing.
Absolutely. Well, it has been so great to talk with you today. It has been just an absolute pleasure, and we love hearing everything you have to say about your research and your background. You can tell you're very passionate about this. It's been great.
It's wonderful.
Yeah. Well, this has been fun. Thank you very much.
Thanks for coming out.
All right.
Thanks for listening to this episode of Conservation Connection.
If you enjoyed our podcast, go ahead and subscribe to make sure you catch every episode that we post.
We'd love to hear from you. So if you want to reach out, go to our website, lastchanceendeavors.com/contact, and shoot us an email.
We love questions from our listeners, so if you heard something that you want to know more about, be sure to let us know.
If you've got a minute to spare, leaving a 5-star review on Apple Podcasts will help other conservation-minded people find the show. We'd really appreciate it.
A big thanks to the people working to protect our planet, and a big thanks to you for listening. Don't forget to tune in next week.